Thank you so much, Dr. Sipe, for that very, very nice introduction. I feel like I have a lot to live up to now. So I'm really, really excited to be here. I am stepping out of my comfort zone, even though I am going to be talking about science. I'm going to be talking really about some interesting ethical dilemmas that have been happening in science in general, which maybe some of you have read about. But before I do that, I need to start out by way of introduction, because it seems like, so how many are first-year students again? Okay, and I have one more question. How many people are declared science majors? Excellent. All right. Okay, that's good. That gives me a feel for who's in the audience. All right, so let's get started. A first question may be one that you've asked yourself. And so just because we can, that is faculty, entice you to come to this lecture to receive maybe LBC credit or extra credit for the couple of you who are here from class, or maybe some honors credit, should we do it? Right, you're here, so hopefully now you have a good reason. And so I hope that the answer is yes. So I hope that there's some value added beyond the credit and the goodwill that you're fostering with the director of the Honors College. I hope you actually learned something tonight, or at least leave with some questions that you'll be inspired to go seek out the answers to yourself. Okay, so you can tell me when I'm done if that in fact is the case. All right, so let's look a little bit Okay, so let's look a little bit deeper into the question that's the theme of this semester's STAR lecture series. And so if we look at the question, we need to think about we. So who is the we for tonight's lecture? And so I've already alluded to this, but the we that we're going to be talking about are scientists, specifically biomedical researchers. So this controversy or this ethical dilemma that I've talked about is really sort of centered in health-related research. And so also this relates to policymakers, and this is where, as you'll I hope get to by the end of the talk tonight, this is sort of where you come in, because at some point you are probably going to have to make some kind of decision regarding what's going on in these ethical dilemmas. Okay, so what is the we doing that is questionable? Well, there is sort of a newish kind of research in the biomedical area that's known as dual-use research. And so I'm just going to throw that term out there right now, and we'll come back to it later on. Okay, so biomedical scientists engaging in dual-use research. All right, so again, ultimately what I hope this will do for you tonight, the reason why, or the take-home message is that you can begin to think about both sides of this potentially high-stakes ethical issue. Okay, so I wasn't actually able to attend Dr. Coolidge's lecture, but I watched it on iTunes U. I thought it was incredibly cool, very well delivered. One of the first points he made was, when you read this question, just because we can, should we? Isn't your knee-jerk response to say, well, no, right? So the question seems a little bit loaded. And so let's overanalyze it. So let's think about what we're asking here. So when you think of this, if someone said this to you, just because you can, should you? Well, and this is not a rhetorical question. What does that bring to mind? And so I'm hoping to sort of engage you guys in a little bit of question and answer here. So what does that mean? What are you being asked? Yeah.
What the catch-22 of issue is?
Yep, I like that. So can you flesh that out a little bit more? Can you think of possibly an example, or can someone help her out? Yeah.
Just because the ice cream looks good doesn't mean you should eat it.
Excellent analogy. I like that. What's that?
Just because all of my friends are doing drugs and drinking doesn't mean I should.
Right, just because your friends are getting drunk or drinking or doing drugs and drinking doesn't mean you should. And so each of these cases, so what is the first part of that? Just because X. So just because somebody's doing something. Now, is that a particular value judgment, or would you, if you were a scientist, would you classify that? I'll make this multiple choice for you. A judgment, A, a judgment, B, an observation, C, a conclusion. What do you think? Judgment, observation, conclusion. Just because I can eat ice cream. Right. So it's more like, okay, so it's more of an observation, or it's an ability, I would argue. So that's sort of where I'm going with this. So maybe just because we have an ability, should we use it? Okay. So what do you guys think about that? So this is my opinion. So this is sort of how I rethought this. So based on our technical ability or intellectual capacity alone, because the question actually says just because we can do something. So exclusively based on our ability to do something, should we do it? Just because I can jump off a bridge, should I do it? The second part of this is a value judgment, right? The should is subjective, right? Maybe you think it's perfectly acceptable to get drunk and do drugs. You certainly have the ability, perhaps if you have access. Probably not here at Eastern, right? But at some other universities. So can do it, the ability, should do it, a value judgment. Do you buy that? What do you think? Yeah, that's okay? All right. So let's proceed from there. Okay, so there's that. All right, so again, where we're going with this is we're going to turn this around and aim it at some scientific issues. Okay, so let's reframe this in the context of research. And so I want to sort of see what you guys can tell me about why you think investigators perform research. So people like me go to years and years and years of school, and we get trained to have the ability to do research, right? And so the question is not should we do it. Of course we should. We went to all this school, right? We have the ability. We should. No, why should we do it? Why do people do research in any field? Knowledge. Sure. So
to teach others.
Sure, absolutely. So to teach others, to expand knowledge, to discover new things, absolutely. What do you think? To utilize it. What do you think?
Pursuit of passion.
Absolutely, absolutely. All these reasons. What else?
Developing technologies.
Yeah. What do you think?
Money.
Sure, right? To improve one's own status. That's a great one. That's a great one. It's absolutely why I do it. What else?
Fame in some cases.
What's that?
Fame in some
Yeah, absolutely. The Nobel Prize, right? Which was just awarded, what, late last week, earlier this week? Yep. All right. So yeah, I was sort of thinking along those lines. I particularly like the fame part. Okay, so specifically, scientists often want to test a hypothesis, right? And so a couple of people said this. Maybe that hypothesis involves a very specific problem to address, or maybe going off in a new direction to create something new. Okay, so a more general sort of purpose is to advance a particular field, and others alluded to this. So it could be biology, it could be chemistry, psychology, education, whatever. But really what you're doing when you're advancing a field is, and I don't mean to oversell this by using the word inspire, but you really are sort of allowing, through intellectual inspiration, others to ask other questions. Okay? So, I'll give you an example. So, when I'm sitting in my office thinking about a research project, I have an office with no windows, so it's very easy to stare at the walls, right? So, strangely, I don't find inspiration there, though. So looking at my empty walls does not help me. I have to look at what's known in my field to try to find inspiration. So I read the literature, and I look for problems to solve or nifty ideas that I can think of sort of a new direction to take from them. Okay, so this is a great reason to do science. And so just to sort of take that another step, I guess I want to sort of graphically depict the scientific enterprise. And so what's really needed to advance a field? And so I guess this diagram is meant to kind of tell you what researchers here at EMU, University of Michigan, at large research institutions do to sort of proliferate what's going on in any kind of field. So everything begins with an idea. From the idea, a scientist will gather some data, because that's what we do. So we form a hypothesis, we gather some data. And then we can't just hang on to that data, because that doesn't do anybody any good. We might as well not have gathered the data if we're just going to hang on to it. So we have to publish it, right? And so after we publish it, usually we need some money, because it costs money to do research, right? So it's not just about wealth and fame, it's about buying test tubes and the things you need for research. So if you can convince somebody, look, I published a paper, I don't hide my data, I really put it out there for other people, often you can get grant money. And then with the grant money, what do? You gather more data. And around you go again and again and again. If you were fortunate, someone will pick up on your publication and think of a new idea. Or maybe your work will generate a new idea, or maybe you'll generate a new idea from your own work. But this really is how the process works. So an important thing to remember, if any one of those squares in the middle is removed, then the whole process falls apart. So money is really important to the scientific enterprise. One has to be able to gather data, or else you can't publish. And if you don't put your work out there, then nobody's ever going to have a new idea. Okay. So questions about the scientific process seem to make sense? Okay. Notice my new light bulb is newer and my old light bulb is. I worked really hard on that. Okay. Yeah. Well, I sort of made this point. But anyway, this is all about, and I guess the phrase that I really want, if you're writing notes, the phrase that you want to write down is free exchange of ideas, because that's really what the scientific enterprise is based upon. Okay. So I said publication is incredibly important. And for those of you who are just starting your college career, it may be that you've never taken a look at a scientific paper. And that's okay, there's still time, don't worry. So let's talk about what's in a publication. So for you more senior students, what can you expect to find if you go to the scientific literature? And we'll talk more about what the scientific literature means in just a moment. You pick up a paper. What are the components of any research paper? Yes, yep, what's that? Sure, exactly. So you see the methods, which describe what the researchers did to get the data, in such a way that if you have close to the same expertise, you can go and do the same thing. That's really key. So what else is there? What's that? Yep. So they talk about the sample. And so if you are doing survey research and you're looking at a broad sample of a certain kind of population, then that will be how you sample that population will be described in detail, right? What else? Yes.
Why it's important, like why are you doing it?
Exactly. So before any paper starts, usually what the authors will do is describe a couple things. That is, why in the world should anybody read their paper? So what's new here? What's useful? What's the new idea, right? Okay, and so after that, then they present the results, so what they did, and sometimes that's mixed into how they got the results. So those are really the components. And across disciplines, that's what you're going to find. They may come in a different order in chemistry as opposed to psychology, but all those things are going to be there. And so the very last point, which I made just a moment ago, is quite key to tonight's discussion. When you find a paper in the scientific literature, everything should be there for you to replicate the study. Okay. So let's talk for a minute about how a paper makes its way into the scientific literature, because it's not like publishing something on the internet. So, which by the way is publishing, but publishing in the scientific literature is very different because it undergoes a process called peer review. So what happens? Well, a scientist gets to a certain point in his or her project. They think, okay, it's time to write up part of this and send it so the world can know what we're doing. Think about it, maybe give us some feedback, some ideas, or have some new ideas that are related to our work. Okay, so the author writes up the paper, sends it to an appropriate journal. There are scads of different kinds of journals in all different fields, so you have to make a judicious choice about what kind of journal to send it in, whatever, so the author sends it to a journal. And so then what happens is the manuscript goes out for review. So it's sent out to experts in the author's field, and it undergoes a single blind review, which means that the reviewers know who the author is, but the author never learns who the reviewers are. So the reviews come back to the editor, they're advisory to the editor, and so Then the reviews go back to the author, the editor makes a recommendation, either sure, let's publish it, no, this is horrible, it'll never fit in our journal, or maybe you need to do some revision, maybe a couple more experiments, and then we'll take your paper. So you get that recommendation, the author does his or her thing, fixes the paper, and then it gets published. So someone has usually three reviewers, have very carefully scrutinized the work before it appears in the scientific literature. That's sort of the hallmark of scientific publication, peer review. Okay? All right. So most of the time, you can believe it. All right, so let's get back to our story. So that's enough in the way of background of science. So just because we can, I guess I want to talk to you a little bit about, or I guess give you 4 examples of some interesting occurrences in biomedical sciences in the last 11 or so years. So in 2001, it turns out that folks engineered, Australian research group, engineered a mousepox virus to be even more virulent than it already was. And it was actually sort of a serendipitous discovery. Okay, Turns out in 2002, researchers were able to reconstruct the polio virus. Anybody get a polio vaccine? So in 2005, very interestingly, and we're going to talk in more detail about this, some researchers in the United States were able to recreate the flu virus that was responsible for the 1917 to 1920 pandemic. This is the Spanish flu. So they actually recreated this virus. And so finally, most recently, if you read the abstract for tonight's talk, this was sort of the teaser. This year, 2 papers were published that described some researchers' efforts to mutate a virus, a bird flu virus, so that it could be transmitted between 2 mammals, 2 ferrets actually, via the air. So bird flu is not typically airborne, but these researchers made it airborne. Okay. So just because we can, is any of this a good idea? I mean, this is bad stuff. Recreate the polio virus, recreate the virus that caused a pandemic, killed millions of people. Cool. What a great finding. I mean, what do you think? Yeah? I mean, do you think? Yeah. Huh? Okay, so I guess the point is, do you think it is a good or bad idea? I see. Not that you understand my point, you understand their point. I'm with you. That's a great point. What would be the point of doing such a thing, right? Great point. Yep.
Well, with all these, they seem to be disease-related, and if you can recreate them, you can build immunities up in your system by introducing a little bit in there. So there's obviously that, so it doesn't become an epidemic again.
Yeah, so hang on to that for a minute. So the point was that it might be that if we knew more about the diseases, there might be a way we could turn this into some kind of therapeutic, right? Is that sort of a fair rephrasing? Why should we not do this? I mean, you might say, sure, what's the point? What's the point of creating something that's obviously not a good thing?
You know, accidents happen, and I really don't want to wake up one day and have the bird flu from the dog.
Right, so yeah, that's a great point. So accidents do in fact happen. So, whoops. So yeah, so there are a lot of bad things that could potentially happen. And I guess let's hold off a minute and address those. Why don't we talk about the example of the flu pandemic for a few minutes. I'll tell you a little bit about the science. I have to tell you, as Dr. Slipe said, I'm an organic chemist, so I have a cursory knowledge of what happened here. So you're welcome to ask me questions, more detailed questions about the genetics. I may punt you to the biology department, but I will do my best to answer them. Okay, so let's talk a little bit about what happened, and then let's get to your question about what the point of this was, and what are the ramifications of the research, right? Because accidents happen. That is absolutely true, and a concern. Okay, so here's the backstory. Spanish flu killed 20 to 50 million people worldwide. It's not, I mean, that's quite a range of people, but records weren't so good then, so it's really not known how many people actually died. Many, many people. A third of the world's population, it's estimated, was infected. This was very serious. So Other numbers have said it killed more than 3 times the number of people that were actually killed in World War I. So it turns out scientists today believe it was a particularly virulent strain. And so certainly in 1917, 1918, healthcare is not what it was not what it is today. So certainly sterilization methods were not practiced in anywhere near the same way as they were, obviously. So you can imagine that transmission of the virus was quite widespread for those reasons. Putting all that aside, researchers still believe that the virus was incredibly virulent. So it wasn't just the fact that it was the 1917 to 1920, it was a bad bug. Okay, what was also kind of interesting about it is, you hear in the news about flu today, when people die, they're typically very, very young or very, very old. And so, pull my laser pointer here. This is my mortality plot of a typical flu. And so this is percent mortality, so how many people die versus their age. So you see right in the middle, young people, 15 to 35 to 40 to 45, you're probably going to be okay if you get the flu. If you're really old, not looking so good. If you're really young, that can be tragic as well. So what was really interesting about the 1918 flu is more of a W plot. So people your age, not so good. Not so good. So it's one of the few instances of flu that really hit the 15 to 35-year-old category very, very, very hard. This was sort of perplexing. And so notice how that fell off, and then it went up again toward the elderly range. And so there's been a lot of research about that, or I guess a lot of conversation. Not so much in the way of research, because again, record keeping wasn't so great, and it's been very difficult to sort of figure out what happened. Turns out there was another fairly serious outbreak of flu about 40 years before. And so it's believed that people that survived that outbreak had some immunity to this particular bug and were able to live. And so they would have been about 40. So everybody younger than that fell victim. So just one operating theory there. Okay, this is just sort of FYI. I found that interesting. Okay, so very bad bug. We don't know much about it, right? But there are worries today, I'm sure you have heard from listening to the news, that there are always concerns about flu pandemics. Okay, so someone said earlier, one way that we can think about sort of mitigating a flu pandemic is to study a flu that would have the pandemic potential. Okay, so collaborators from the Centers for Disease Control here in the United States, the Armed Forces Institute of Pathology and Mount Sinai Medical School got together and said, let's recreate this virus and see what we can learn about it. Well, it had to be recreated because there was not a living sample. And so what they in fact did, and they managed to regenerate it in 2004, is they actually got a tissue sample from someone who was buried in the permafrost in Alaska. And so they were actually able to get the genetic detail from that sample and use a technique called reverse genetics to make the DNA, and then take the DNA to make that into the virus, inject it into live cells, and make it into a living virus. Okay, then they put it into mice. And so with apologies to the mice lovers in the room, we'll go on to the next slide. Okay, so this is a cartoon of typical flu. So anybody know how many genes humans have, ballpark? You want to take a guess? Higher than or lower than 100? Not quite millions, but you're, a lot. Yeah, any idea? Smart guy in the front row. So roughly 20,000. I'm sorry, that's what you were going to say, right? Roughly 20,000.
I was going to say hundreds of thousands, but
Base pairs get you. So roughly 20,000. So flu, 8. And they're not round and they're not colored, but 8 genes for the flu virus. So it turns out, in the scheme of making things, not that difficult because there are just 8 genes. Okay, so here's the typical flu virus. If you stick it in a mouse, it gets sick and it coughs and sneezes, right? But it's okay, it lives. So the flu virus that this CDC researcher actually wound up sticking this into a mouse. And so what happened Very bad. So the mouse died in 3 days, which is, yeah, I'm sorry, it's just a cartoon. So what was really remarkable about this is it was just, it really knocked the mice out, devastating. And usually, you know, so it was certainly the fastest annihilation of mice for any flu strain that had been tested to date, by far. And so it's kind of amusing. So the guy at the CDC, the scientist at the CDC, you know, was interviewed after this, and you'd be thinking, oh my gosh, it just knocked these mice out. Wow, oh man. So here's what he said. I love this quote. I was quite surprised. I didn't anticipate that they would die that quickly. That was really quick, right? It's a horrible virus, right? Killed the mice in 3 days. I was quite surprised. Okay, scientists notoriously understate things. All right, so they weren't done there. So here's what's arguably kind of cool or really scary, depending on how you want to look at it. They, in fact, made this thing. Okay, so that's the point here. And so what they could then do is try to figure out what made it so bad. And so what they did is they took the typical flu, and so they switched one of the genes, the purple one, with the 1918 flu. Bad news for the mouse. So it killed the mouse, again, 3 days. Then what they did is the reverse experiment. So they took the 1918 flu, switched the purple gene with the regular flu, and you guessed it, mouse coughs and sneezes but lives. So they could actually learn something about the cause of this excessive virulence. Pretty nifty. Okay. But scary, right? Because now we know the DNA sequence for all 8 genes, right? And so you guys have heard of the Human Genome Project. So we have like the virus genome for the 1918 flu now. And so what this will allow us to do, allow immunologists to do, is to probe more deeply so they know what gene is responsible for this extreme virulence, so they can actually look at the DNA for that particular gene, learn more about it, and perhaps design new targeted therapies related to this. Cool. Maybe even mitigate a potential pandemic. But now we have live virus of the 1918 flu. Well, we didn't before. And so sure, it's sitting in a secure location, but there can be lab accidents. I mean, it has this high security around it. But still, there are safety concerns. It would have to be some kind of incredible heist, but there could possibly be theft. More importantly, they published this in probably one of the 2 most prominent science journals, the journal Science. And so every detail is about how they made this is now in the literature. What's in a paper? Everything you need to be able to do this. Maybe not you, because you don't have a PhD in immunology, but if you had the right expertise, you could recreate this and the right equipment. So the information is out there. What do you think? Good thing to do? Bad thing to do? Yeah.
It's neutral. It has no morality. It depends on what the individuals choose to do with it. I mean, it's akin to when the atomic weapon knowledge or thought upon how to develop it was illegal at a point.
Yep.
Even if you didn't have access to it.
Yep.
I'm a lot less philosophical than that. In my mind, if the literature wasn't out there, I wouldn't be so terrified. Because as long as it's well protected and we use some logic, it's no big deal that, yeah, great things can come from it. But when you put it out there on how to remake it, I have these flashbacks to evil scientist movies, and it's not good.
Yeah. So certainly, it's a tool. Like I make reactions in my lab that can be tools for other chemists, right? But the tool can be used for good or it can be used for nefarious purposes. Certainly true. Absolutely true. It's all in the mind of, I think that's really well stated. Depends on the mind of the user. Yeah.
I think at the same time, though, you have to focus on, we learn from challenge and change. We learn from production. We learn from enhancement. We learn from advancement. Without all of that, we wouldn't have the technological findings that we have
Mhm,
So
mhm.
we
Yeah.
have to take a risk or we'll stay stagnant and die off as race.
So you're sort of at the heart of the argument, right? So do we do this work and put it out there and let people have new ideas? Do we take an idea and let it go through the process and spawn new ideas, which might lead to potential cures for pandemics, right? Or do we put it out there as a potential bioterrorist weapon, right? Yeah. It's a big question. It's a really big question. Okay, so here's what we've just said, right? So public health and safety, the potential of a bioterrorist threat versus advancement of science. But advancement of science contributes to public health. Interesting, right? So it's public health and safety versus public health and safety. It's a difficult question. So shall we push on, or do people have thoughts at this point? How are we going to solve this, right? Huh?
Kind of a cause and effect, what would be the cause versus the effect? I mean, could anything happen? Sure. But I think it outweighs, the benefit outweighs the risk.
Right. So that's an interesting point. So how real is the threat? Yeah.
I like how you stated how it's a simple tool, it can be used for both good and bad. And just from my own thought is, yeah, there's some bad things that have come out of creating a tool like a knife. But it seems like there's been a lot more good that came out of it than just the bad.
Right. We need to cut things a lot.
Now,
Yeah.
only is the bad good,
Sure. Sure.
but what level of we should.
Yep. Yep. You, then you. Yep.
I think that, like, if the results couldn't be generalized, it wouldn't be worth it, because then you're just creating the cause just to find, like, the effect. Like, if the only applicable, I guess, cure is just for the 1918 flu, like, there was no reason to bring it back. But if by studying this flu, you can learn things about other flus, then I think it's a good idea.
That's a great point, and one that I forgot to put on a future slide. So keep that in mind. So when you're weighing the relative goods, you really have to think of the scope of the threat, right, versus the scope of the potential payoff for the scientific research, the potential application. That's a really interesting point. Thanks. Yep.
I think when you're weighing the costs and the benefits, I think it's worth it to advance science for public health, even though there might be a public health concern, because whatever advancement it is going to eliminate the concern that was there. Well, may eliminate the concern.
Yeah, that would be the hope. That would be the hope. Yep, that's true. Yeah.
And even though like this was seen as something potentially dangerous, sometimes something that was intended to be a purely good purpose could be used by somebody else in a bad way. So it's like you can't.
That's great. That's great. That's exactly right. Yeah.
I'm not sure exactly, I can't remember the exact name of the mathematician,
Mhm.
but there was, in my calculus class, we talked about a mathematician who was trying to decide if he should believe in God or not, and he put mathematical values to it based upon the consequences of his lifestyle if he lived it a certain way and if he chose to live the other way, and then using those numeric values he put to that, he decided that he should believe in God, and it was just an interesting kind of relation that I thought.
That's very interesting. It's been a while since I was an undergraduate. I have a vague memory of studying that myself. Yeah, and we're going to talk in a minute about so this problem hasn't gone unaddressed, right? And we're going to get to that in a second. And so it's been really interesting to see sort of the rubrics, right? The value structures that people have come up with to try to walk their way through these kinds of ethical dilemmas, not unlike the mathematician. They haven't been so mathematical about it. But yeah, I mean, it's sort of interesting what people come up with to try to draw a line where, really none exists, in my opinion. So other comments before we push on? This is great. Keep talking, y'all. Nice. Okay. Okay, so in academics, what we do is we form committees. And so that's exactly what was done in this case. So a committee was formed in 2005 called the National Scientific Advisory Board for Biosecurity, or NSABB. If you Google that, you can go to their website and read a little bit more about this if you're interested. And so what they do, as the name implies, is they advise the US government on sort of what to do in these situations. And so they consist of 25 voting members. Mostly they are research scientists at public, and that means state institutions that get a fair amount of money from the federal government. Okay, so places like University of Michigan, very large research institutions, some private research institutions as well. There are a couple of attorneys. There are some people that represent workers in scientific fields, so their interests are represented. There are also 16 ex officio members, and so those members represent every office of the federal government that has a stake in this ethical issue. And so you can look on the website and see all the offices, and it's sometimes sort of surprising who has their finger in biomedical research, but it turns out a whole lot of offices do. Okay, so actually they weighed in on this science paper publication that I just described to you because it was going through the publication process right as the committee was formed. And so it turns out that right as the editor was making a decision, he sent it to the committee and said, hey, you on board with this? The committee was a little bit miffed that they really didn't get much time to think about it, but they eventually signed off on it and out it went. Okay. So let's talk about this committee's recommendations. And so they have come up with, actually this term predated the committee, but this type of research that we've been talking about has come to be known as dual use research. And so dual use research is research that can be used for beneficial goals or malevolent purposes, right? And so second bullet point, just my opinion, I think this is a great marketing technique, dual use research. Do you want to do dual use research? Absolutely. Research that can be used for two purposes instead of one. Sounds great. A whole lot better than research that could be weaponized. So anyway, what the committee also did was to challenge scientists. So they weren't in the business really of making mandates, but really what they want to do is to challenge scientists to be aware of potential dual use issues and also to be responsible once they're aware that those issues are there. And so the responsibility really means, okay, you know you're doing dual use research, then you need to organize your lab, educate everybody as to the protocols, the protections you're going to take, that kind of thing. Have a point person in your group who's going to sort of shoulder the responsibility of dealing with all the paperwork and that kind of thing. Okay, so dual use research. But how do we define dual use research? And somebody hit on this a little while ago. The person who mentioned a knife, right? Just about anything can be considered dual use. Take a car, for example. Unfortunately, a car can be used as a bomb, right? But we all drive them. So a car can be dual use. So where do we draw the line here, right? So fortunately, the committee addressed that. However, it's a bit of an arbitrary line. So they decided that they would reframe this dual use issue into dual use research of concern. And I'll let you read this. I might as well read it to you. So it's research that, based on current understanding, can be reasonably anticipated to provoke knowledge, products, technologies that can be directly misapplied by others to pose a threat to public health and safety, agricultural crops. So if crops were destroyed, other plants, I left out the T there, animals, the environment, or material. And so even something that could possibly devastate the economy would fall into this category. Okay, so I want to sort of boil that down for a minute to sort of hit the key features. So let's really sort of think about what type of research we're talking about. So the key features, based on current understanding. So if you're doing research that today you can imagine could be reasonably applied as a bioterrorism threat, then that's dual use research. It's not, you don't want to take a science fiction approach. If then that, if things that aren't even conceived of happen, or aren't realistically in the literature could happen, then maybe this could be a threat. No, that's not dual use research. That's not dual use research of concern. Okay. reasonably anticipated. So another if is how much expertise would really be required to create a threat from a paper. Would someone have to go out and find 4 scientific papers and synthesize the information from all 4 of those papers together to make a threat? That's unlikely, right? If all the information is contained in a single document, in a single paper in the literature, yeah, that's probably dual use research of concern. Or if it's not much of a jump to find another piece of information to put with it to make it a threat. Okay, finally, not an isolated threat, very ambiguous. So imagine a car as a bomb. Very tragic situation if that occurred. That's kind of, unfortunately that would just be a handful of people. That's not a pandemic, not an isolated threat. And I'll give you an example from my discipline, from organic chemistry. So about 10 years ago, people were very, very interested in making molecules called tumor promoters. They do exactly what the name implies. They promote cancer. And so people were really interested in studying them, and so people built them and made modifications to them to see if they'd be better or worse at promoting tumors. Now, it would be really unfortunate if someone stole a tumor promoter and harmed someone with it, but it would only be 1 or 2 people because these things were made in such minute quantities. It's not something that could, hurt a mass of people. Okay, so not probably broad enough scope to be considered dual use research of concern. Care would definitely still need to be taken, of course, but it would not fall in this category. And so it wouldn't fall under the kind of research that would require some serious, I'm not going to say regulation because those have not been enforced yet, but serious oversight. Okay, so are we sort of clear on the kind of things we're talking about? And let's go back, oops. Yeah, so every example that I showed you here really is dual use research of concern. So in the first case, what folks are doing is they're enhancing the virulence of an existing virus, right? So this has obvious nefarious potential, as does the reconstruction of the poliovirus, certainly the 1918 flu virus, and finally, the airborne version of a flu virus that I'm going to talk to you a little bit more about in a minute. Okay. Oops. Oh, and so we talked about this, not really dual use research of concern. Okay, so the last point I want to make, and this is really where things sort of get sticky. And so, like I said, this committee is not into making mandates, but what they are interested in doing is making recommendations for scientists to think about how they disseminate, i.e. publish their results. Okay? So, what kind of content would you put in your paper if it were a dual use research of concern paper? Would you publish everything as is? Right? Would you make details available on request, so make the information sort of classified? Or would you withhold it altogether? Right? And so the idea is federal agencies will make recommendations, and in principle, you will follow their recommendations. So again, these are not mandates, but it's sort of generally accepted that scientists will follow these rules. Timing, so do you publish now, or do you wait till a later date where maybe the threat is mitigated? Finally, who's going to see it? So do you publish it widely in the literature? Again, can you make it classified? Or do you not distribute it at all? What happens if you don't distribute it at all? Well, then we don't have publication, and so there's a breakdown, remember, in that cycle of research. Okay, so we've got these recommendations, but the problem's not solved. So there's one issue with the committee straight away. It's made up of people in the public sector, universities, whatnot, not really anybody representing the private sector, nobody outside the United States. So it's all fine and good if we at public institutions follow these recommendations. Well, what's the rest of the world going to do? And what are the folks at Pfizer going to do? And at the drug companies, where there's lots of money tied to this. Are they going to follow these recommendations? Well, there's nothing right now to make them do that. And you know, maybe there shouldn't be. Okay, so again, they're just recommending guidelines. They're not making mandates, so there's really sort of no teeth to this committee. And remember, there are consequences associated with each choice. So here's sort of a graphic of what I just said. Remember this. And so if we restrict publication, bang, right? So what does that do to scientific enterprise? Okay. All right, so the last thing I want to talk to you about is to tell you one more story. This one's shorter, about what's been happening recently in terms of the development of an airborne version of avian flu. Okay, so avian flu, which is a virus that is not transmitted from species to species. So it doesn't cross species typically. In rare cases it does, but it's not transmitted from birds to people. So the way people get it is contact with infected birds or secretions from those infected birds. Fortunately, once a human is infected, it's very, very bad for that person, but it typically is not transmitted from human to human also. So that's good. Usually it's possible that there can be maybe one transmission, but beyond that, that's it. The bad news is it is quite deadly. So it's known as H5N1. So researchers asked, you know, what if it can mutate, and then, you know, what if it could actually transfer from mammal to mammal? Again, that could cause a pandemic. That would be a big problem. And viruses mutate all the time, so it's not completely unrealistic to think that. And then, well, we do, right? So the idea, engineer that virus and take a look at it and see if we can figure out what we would do in such a case. So people use cute animals to study these kinds of things. The ferrets lived, that's a ferret. The ferrets lived in the story, so that's good news. Okay, so there were two research groups just in the past few years, one at the University of Wisconsin in Madison and another at the Erasmus Medical Center in the Netherlands who were working on this area, basically simultaneously with two different sets of experiments. So essentially what they did is they either mutated or combined avian flu with swine flu and made it so it could be transmitted from mammal to mammal. So they tweaked the bird flu, they injected it into ferrets, and then they tried to determine whether, or they determined rather, whether one ferret could transfer it to another. Turns out it could. So they actually made it airborne. So the good news was that no ferret that was infected with an airborne virus actually died. So it turns out that in the course of tweaking the virus, they actually made it less virulent. So that was, you know, sort of the silver lining. Yeah?
Except with the lack of information that we have from this, for all we know, ferrets might have a natural resistance to the
Right. So, yeah, and so this is a very controlled environment also. So that's right. It's one of a series of experiments. You're absolutely right. Yeah, so I don't know if you heard the comment here, and to paraphrase, you know, that tells us very little, I guess, is sort of, yeah, what you're trying to say. And that's true. That is exactly true. But it begs other experiments, right? So, you know, what can we, or, you know, it says, look, we just made an airborne version of the bird flu. Is this a good idea? So the controversy began. So now we have this committee, right, that I told you all about. Clearly, this is dual use research of concern, right? And so committee springs into action. And so the controversy ensues. So both of these groups that I told you about that are working on this are funded by the largest funding agency in the country, federal funding agency in the country for biomedical research, the National Institutes of Health, huge funding agency for biomedical research. And so the NIH goes to the committee and says, you know, what do you think about this? And they say, you know, let them publish papers, but let's take out the methods section. So they were all about redacting the methods section, so no one could reproduce it. And so that is absolutely one option. But then, interestingly, the results came out. And the results were, as I just told you, that the ferrets didn't die when they made this airborne version of the virus. So the committee changed their mind and let them publish the papers in full. And they actually just came out May-ish. One was in Science and one was in Nature. And those are the two most widely read journals in all of science. So they're out there for everyone to see. Interestingly, at the same time, both groups imposed a voluntary moratorium on the research. And that's been going on actually since January. And so they're holding off, they're not doing any more work until there can be some kind of international agreement about, first, biosecurity and containment of this virus. And also, something that the committee is trying to get the scientists to do is to better educate the public as to why in the world we want to explore this. And so we can have this conversation, and together we can think of good reasons why this research can be valuable, right? But this is sort of where you come in. This is the kind of thing that you should think about and certainly examine both sides of. Okay. So, last couple of points. So, for dual-use research projects, you really have to think about current threats versus potential outcomes. For you personally, you need to try to stay informed about each side of the issue. And why? You're not a scientist right now, you're an undergraduate, right? Why is this important to you? Well, you can vote, right? And so, as I just told you, this kind of research is paid for by federal dollars. So the president and Congress decide on NIH's budget, and you vote for the president and Congress, right? So really, this comes down to you. So it's something that you should certainly be aware of. And at some point, right now, this committee is sort of in its infancy, and it's certainly possible that congressional oversight could get involved, and then things could get really interesting. Okay. So we can do lots of things. So our intellectual capacity affords us the ability to explore all kinds of things, right? So should we? Yeah, maybe, right? But we need to consider consequences. We need to consider the consequences of pursuing these things, take proper precautions, or we need to consider the consequences of either pursuing and not publishing or not pursuing at all. So that's sort of where we are on the issue. Okay.